Okay, our system of taste, our gustatory system, works in a similar way, but is also quite different than our taste of smell, than our sense of smell. And so we'll think about the different tastes that are out in the world. And the reason we need to taste is to assess the amount of nutrients that are in the food that we're going to eat, and whether it's the nutrients that we want, and whether there's any poison or anything like that. And so we'll see how the brain accomplishes this goal. So our gustatory system, our primary receptors of taste, are taste receptors that are in our tongue and a mouth cavity. And these taste receptors are organized in clusters, in these round onion-looking groups of cells. And you can see that each of the cells has little cilia, and that those cilia extend out into this area called a taste bud. And so this group together that looks like the onion is the taste bud, and their little protrusion here is where they can collect all these chemicals in the saliva. But notice from this image here that these taste buds don't come up onto the top of your tongue, but in fact, whenever you have something very sour or salty, sometimes you see bumps on your tongue. These bumps are these little papilla, and the papilla on their sides have these little crevasses, and in the crevasses is where these taste buds are. So these taste buds line the walls of these crevasses, and the saliva gets sucked into these crevasses and then can activate the receptors of these taste buds. So the receptors themselves are embedded into these cells, and each cell has only one type of receptor, but the cells are clustered together in these groups of 50 or 100. And it's interesting because each of these 50 or 100 cells doesn't have its own axon. In fact, the axons leaving each taste bud receive information only from a single presynaptic cell. All the other cells in the taste bud, even though they have different receptors, they combine all their information and send a signal into the axon. So the taste bud acts as kind of a processing unit together, and depending on the ratios of the different types of taste that can come in, different types of chemicals that can activate these receptors, the output of a given taste bud will be modified. And so the sensory neurons downstream of these taste buds receive information from lots of these receptors. One misconception about our gustatory system that you likely hold yourself is that there's certain locations on our tongue that have different types of receptors. So you see lots of images like this where there's an area on the tongue for bitter detection, an area for sweet and salty detection. In fact, we now know that we can investigate this in more detail, that the tongue has different types of taste receptors scattered throughout. So there's no dedicated zones for sour or bitter or salty or sweet. We have 5 tastes that our tongue and our mouth seem to be able to be responsive to. You've likely heard of 4 of them quite a bit: sweet, salty, bitter, and sour. And we have a 5th taste, umami, the savory, meaty taste. And each of those different receptors is activated in a different way. Some of them are quite simple. For instance, the salt and sour receptors are just open channels that let in sodium or let in hydrogen. And when they do that, they change the internal equilibrium of the cell, and they bring it closer to or further from firing. Whereas you can see that the sweet, bitter, and umami receptors are G protein coupled receptors. So they start, once the key is entered into the lock, they start a cascade of processes that ultimately releases calcium and increases the firing of the cell. So either they directly let in the chemical that they're sensing, or the chemical is detected like a neurotransmitter and starts a cascade of processes that leads to depolarization. But in all cases, the cell then can either send or not send signals to other cells. Remember that in this onion group of cells in a taste bud, a lot of the cells communicate through gap junction. So they're not sending neurotransmitter from one cell to the other. They're sending signals across gap junctions in their cell membranes. Interestingly, we have 30 different types of receptors for different bitter tastes, which also says a lot about the evolutionary value of being able to detect a bitter poisonous substance and not eat it. How about the pathways from the mouth into the brain? So the afferent nerves that are admitted into the nervous system from the gustatory system leave the mouth via 3 of the cranial nerves that we learned of. We learned 12 of them, and 3 of them are the facial, vagus, and glossopharyngeal. Remember that these nerves both carry motor information out to the body and take sensory information back from the sensory receptors into the brain. So you can see that the different nerves pick up information from different sets of sensory receptors, either the front, the back of the tongue, or the mouth cavity itself. And from there, information travels to the medulla in the brainstem, to an area called the solitary nucleus. And from the solitary nucleus of the brainstem, this is an area just like a lot of the other cranial nerves. A lot of the nerves enter into the brainstem before traveling up to other areas, just like they do when they come in from our body. They would go through areas of the brainstem. Then they get to the thalamus here, the ventral posterior nucleus of the thalamus, which is the same nucleus of the thalamus, the VPN, that received information from our olfactory system. And from the thalamus, they project into this top part of the lateral fissure here, which is part of the insula. So remember that we saw that the auditory cortex was on the lower half of the insula here, and our gustatory cortex is on the upper half of the insula. And from there, it's not shown on this diagram, but the information projects to the orbital frontal cortex to meet the information from our olfactory system. So this pathway goes from the tongue into the brainstem to the thalamus to the insula and then up to our frontal cortex. Well, you'll recall is where the conscious perception of the taste and smell is represented. We can get brain damage that can lead to disorders of smell and taste. And one quite common one is suffered when we get a blow to the head. And a blow to the head can shear all these axons that are going through the cribriform bone here and can cut all the axons off. And so you lose all the incoming smell information from all these cells. And so without all these axons, which are receiving all the smell information, you can lose your ability to smell anything. These cells would still be active, but they wouldn't stimulate the olfactory bulb in the same way. So maybe you can think of something analogous to a cochlear implant that we might invent one day that could continue to stimulate the olfactory bulb depending on which odors are detected. I think we have a lot of work to do because we need to better understand how different odor detection leads to different activity in the olfactory bulb. We can also lose our sense of taste, but it's much more rare. And it's rare because there's 3 different axons that take information from our tongue and oral cavity about taste into our brain. One of those pathways, the facial nerve, innervates the front of our tongue. And from there, because of a quirk of evolution, the axon to get to our brain passes through our inner ear. So you can see this axon traveling through the inner ear here on the way to the brainstem. And if you get particular damage to your ear, maybe in a car accident where you hit your head, you can actually lose damage to the facial nerve. And you'll lose the ability to taste on the anterior 2/3 of your tongue, only on the same side as the damage to your ear. So this is called agousia. So we have agousia and anosmia, or the inability to taste and to smell. Okay, so our olfactory and gustatory system's job is to detect chemicals out in the environment. And this can be for two reasons. One, we want to detect chemicals that we want to avoid. We want to run away from noxious odors. We want to avoid eating plants that don't taste good, that have poison in them. And then the other thing we benefit from having an olfactory and gustatory system is we want to be drawn towards things that smell good, that are beneficial for us. We want to taste things that are healthy for us. So we love the taste of melted cheese and butter and sugars because they're very jam-packed with calories. And what we need when we're starving is some very good, strong, and easily digestible calories. And so we also saw how these two systems have different pathways into the brain. We saw that the olfactory system has this direct link into our limbic system, so it seems to be able to evoke certain memories quite easily. And we saw that the gustatory system in the mouth sends information to the same part of the thalamus and also to the same part of the orbital frontal cortex. And it's likely that the information is combined from our taste and our smells in the olfactory cortex to represent this conscious perception of what it is that we're enjoying or avoiding at the time. And this is where information about our hunger and how motivated we are and the look and the sound of food would also come together to represent our perceived flavor and perceived enjoyment or disenjoyment of something. So I'll start one more video and we'll cover attention, the third topic. So I'll see you there.